Okay, hi everyone. So we're happy to introduce today Ming-Jung. So Ming-Jung is an associate research fellow at the ABRC, which is the Agriculture Biotechnology Research Center in Academia Sinica in Taiwan. She did her PhD in 2012 under the supervision of Xing Wu at the EBMP, which is another institute in Academia Sinica. She worked mainly on translation and photomorphogenesis. And she was at the origin of the first paper, one of the first paper, probably the first paper on ribosic data in plant. Next, she did her postdoc at USA Michigan, where she worked on transcription factor and nucleosome positioning on plant gene expression, mostly on tomato. Since 2016, Ming is a group leader at ABRC in Tainan, which is the south of Taipei in south in Taiwan. And she worked on alternative translation and on non-AUG translation in plant. She obtained in 2022 an EMBO Global Investigator Award, which is an award that helps a young scientist from Asia region to settle down and do good research. So,
So thank you very much for the nice introduction and also the invitation that I could be here in this charming campus and have a chance to visit this lovely city. So thank you. Okay. So before I start, I want to first introduce where I come from, as Shangma have introduced. I come from Academia Sinica. That's a top research place that I would recommend in Taiwan. So in Academia Sinica, we contain many different research institutes and also centers. So where I come from is Biotechnology Center in southern Taiwan. Located in the southern is another lovely place in Tainan. So in our center, we focus on agricultural problem. We're also doing some mission-oriented basic research. Honestly, this is confused to me. In my words, I would say we are doing create knowledge related to agricultural problem. So hopefully I could convince you I'm doing that. So in our center, we have several major topics like agrogenomics, where I belong, but also we do small functional molecule like metabolites. And also we focus on different crop species or economic species. So we also develop some biotechnology like CRISPR editing in like OKI. So that's what we are doing. So please check us if you are interested in our research. Yeah, feel free to let me know if you have any question. Okay, let's go back to my research. So I really appreciate the invention of the airplane that bring me to here very easily and make travel easier. So every time when I am in the airport, I always wondering how many runway are there within the airport, and how the airplane to find out the correct runway to land on and to take off. And I think the similar question could refer to the question I'm interested in translational control and protein synthesis. In the cells, there are so many different mRNA molecules. So how do the ribosome know which molecule to bind on and where to start the protein synthesis and to terminate the protein synthesis? So in this talk, I'm gonna tell you one of our lab's research line to finding the hidden genes from the translation perspective. Okay. So one very big news last year, we all know that these two great scientists, because their research in mRNA modification that facilitate the mRNA vaccine development and won the Nobel Prize. So how the mRNA vaccine works? We all know that they deliver a very small viral mRNA into our human cell and producing protein that's the antigen and trigger the immune response. But to complete this process, there's one very key critical regulatory process, that's a translation. The ribosome need to recognize the correct viral mRNA to find out the translation initiation site, then produce to the correct protein. That's for this translational process is very important. They need to at the right time in the right place to produce the right protein. So my lab has been interested in this process. We are interested to know that for a given mRNA molecule, how many protein and what protein could be encoded from one single molecule, and how this translation initiation event is started. So typically in eukaryotic system, usually the ribosome will recognize the 5' end of the mRNA and scan for its initiation site, which usually occur at the AUG codon and produce one protein. So in eukaryotics, most of the translation, we are thinking about one messenger RNA, one AUG initiation site, and one protein. However, interestingly, in the past 10 years, there are more and more evidence has been supported. There are more than one translation initiation site could occur on a given mRNA. For example, we know that there are some hidden open reading frame, or sometimes called genes. So in this talk, when I say hidden, it means that they are not annotated on our gene annotation file. So there are many hidden open reading frames. For example, they could located in the upstream open reading frame in the fram UTR region that could encode some small peptide or regulate the protein expression of the surrounding genes. Or sometimes you may heard people say, okay, this is a non-coding transcript. However, there are small open reading frame on it. They have the potential to encode small protein or peptide. And in some cases, there are some alternative initiation sites that could encode different protein isoform, and they are important to control some physiological response. For example, the flowing time. People know that there's one important gene, FCA. They could be encoded actually from the non-annotated initiation site to have a longer protein isoform. And more interesting, they found out for these genes, they could initiate from a non-AUG codon. In this case, they are initiated from a CUG codon. And this codon, you can see they are conserved across different plant species. And also in some cases, in mammalian system, they also found some mammalian ribosomal protein 18. Depending on which condition they are, they could be initiated from different initiation sites to encode different protein isoform and regulate their heat shock tolerance. So from this data, we know that indeed on the genome, there are some hidden genes or hidden initiation sites. We haven't been exposed systemically, but they are still important to produce some hidden peptide or isoform. They are important. So as I say, although they are important, however, due to the annotation bias, only limited cases has been studied. So when I initiate my lab, we want to address this question. So specifically, we want to know to what extent on the genome are there any novel initiation sites that could encode the novel genes on the genome? So if there are some, so how the ribosome regulates or recognize this novel initiation site? What are the recognition mechanisms? So to address this question, we focus on the plant species Arabidopsis, tomato, but also the plant viruses. Okay. So as we mentioned, so our first question is, we want to know to what extent this novel translation initiation site could occur on genome. So in order to address this question, we use a technique we call it ribosome profiling. So typically, I think most of people we do the RNA sequencing. When we say RNA sequencing, not the total RNA sequencing, we purify whole length mRNA, we fragmented them, and we sequence and map it back to the genome. So you can see that in here the GM model. So based on total RNA sequencing, we could detect the recon signal in the UTR region, exome region, coding region, but not in the intro. So that's perfect. So what's a ribosig? It's just one step further because we want to detect ribosome positioning. So we purify the ribosome associated or ribosome protected fragment. Then we sequence this small mRNA fragment and map it back to the genome. So we could have the information about where the ribosome they bound indicates those regions are under translational process. But since my goal is to characterize the in vivo translation initiation site, so here we perform a photochemical treatment that latinomycin, we call it LTM, which is specific freeze the initiation ribosome on mRNA. So with ribosome profiling combined with a chemical treatment, we could get specific signal from the initiation ribosome. So you can imagine that when we map the reads back to the genome, just right on the annotated initiation site, we could see the LTM signal indicating in vivo translation activity. So please keep this in mind, because in the following, I will use LTM signal to infer initiation translation activity. Okay, so we just perform this technique first in tomato. So here is our data. So if we focus on the annotated genes, this is a plot I just showed you. Again, if we look at LTM data sets, we could detect in vivo initiation activity on annotated initiation site. So we are happy about this at that time. Okay, so it means that our success and our technique is success. We successfully profile the in vivo initiation ribosome. So based on our data set, we further could find out some interesting cases. For example, in another genes, we could identify a second novel, I should say the second in vivo initiation site, just right on the 5' UTR region, which could encode a small upstream open reading frame. And through Webster plot analysis, we found out that yes, this second initiation site really could trigger the small protein production. And in other cases, we could identify, in some genes, we could identify a downstream inferent second initiation site, which could encode an N-terminal truncated protein isoform. So from here, you can Western blot, you can see we could detect two different protein isoforms. So indicated, yes, at least some of the initiation site, we call this alternative initiation site, that really could trigger protein synthesis. So here is the case I show you single cases. But since our goal is to know like globally, systemically, how many genes or how many, to what extent this novel initiation site could occur on genome. So we just performed the bioinformatic analysis and scan gene one by one to find out any novel initiation peaks in vivo. So here's our data. So in total, we could identify more than 11,000 initiation, I will call it LTM peaks. So we infer as a novel, so we infer as an in vivo initiation site in around 8,000 genes. So if you quickly do a calculation, you will find out that actually one gene could have more than one in vivo initiation site. So indeed, we could identify around 30% of the gene have the novel initiation site. So it indicates that on the genome, on the genes, there are many genes, they have more than one in vivo initiation site, have the potential to encode more than one protein or a small peptide. What's more interesting is that we found that if we check the codon on this novel initiation site peaks, you will find out that some of them will locate it on AUG, that's expected, but some of them could locate it on non-AUG codon. For example, in this case, it's a CUG codon. So indicate, yes, on genome, there are some non-AUG initiation site. So since right now we have collected this data set, we further want to know to what extent this in vivo initiation site could occur at the AUG or non-AUG codons. So we go to this initiation site one by one and check their codon sequence. And here's our result. So when we first focus on our tomato in vivo initiation site, that's around 11,000 initiation site. Yes, 50% of them are located at the annotated one, that's the AUG codon. So our data confirm the in vivo translation initiation activity on this annotated one. So that's great. But it also means that the rest half of the in vivo initiation site haven't been annotated. And we could find that the majority of them are located on the novel AUG codon. And the second major group is located on the near concatenate codon. So what is near concatenate codon? Near concatenate codon are the codon just one base different from AUG. So it could be the CUG and the ACG. So from here, we know that there's a second major codon group that could serve as the initiation codon. That's near concatenate codon. And this pattern, we also analyze the data from Arabidopsis, because actually during we develop our project, the Arabidopsis data set comes out. It was scary for me, but luckily the question I want to address, they haven't been addressed in their paper. So we just reanalyze their data. And indeed, we found the same pattern. The second major group are the near concatenate codon could serve as the initiation site. And also it has been found in human. So this data gives us an idea that, of course, eukaryotics or in eukaryotics, they are not only the AUG, but also non-AUG codon could serve as the initiation site for protein synthesis or translation. So now I address the importance of the near congenate codon could serve as the initiation site. But actually, they are just one base different from AUG. So in total, for the near congenate codons, actually there are 9 different codons. So we further want to know which ones are preferred or not. So from the enrichment analysis, you can see generally across eukaryotics, CUG and ACG was most preferred to serve as the initiation codon. So we further found that the codon preference when they are the near congenate codon, actually CUG and ACG are most preferred to be initiation site. Okay, so this is our finding that about the codons. So they are novel initiation site, novel AUG could serve as the initiation site. But how about their physiological impact? Are they really important in biology-wise? So as I mentioned previously, this novel initiation site could trigger protein synthesis, and that could induce the protein expression diversity. And another case is that because we know many organelle localization signals are localized in the protein N-terminal. So if there are some alternative initiation site that induce different protein isoform that could regulate their organelle localization. So for example, in this tomato genes, we could identify a second alternative initiation site, which encode an N-terminal truncated protein isoform. Then we further found that they are predicted to be the mitochondria. So to demonstrate this, here we further clone this 4 coding region contain 2 initiation sites. And indeed, we could find that this GLP signal overlap with mitochondria, indicates yes, there are some protein localized in mitochondria. However, if we just mutate this downstream initiation site, the overlap signal disappeared. So indicating yes, this second novel initiation site could encode a shorter protein isoform to regulate protein localization. And what's more interesting is that when we check their autologous gene in aerodosis, they also show similar mechanism. So indicates that this alternative initiation site that regulates protein localization, this mechanism, actually they are important and at least conserved between DICA in tomato and aerodosis. Yeah. Okay. So this is an example I talk about from the plants. So from our beginning, we start from the ribosome profiling and to pinpoint the in vivo initiation site. So on different plant species right now, we know there are prevalent novel initiation site on plant genes. They could tend to occur at the AUG or non-AUG codon. And they regulate protein not only expression and also their functionality like localization. Okay. So this is our story from plants. However, plants, they are not the only organism to use their translational machinery for protein synthesis. There are other organisms. For example, the plant viruses. So since we could find out some novel initiations that haven't been found on plant genes, then how about plant viruses? Okay. So in Taiwan, there's a very severe disease, plant virus disease. We call it tomato yellow leaf curl disease. Actually, it's not only important in Taiwan, it's also worldwide. So this virus has been ranked as a top 3 due to their agriculture and economic impact. So you can see that when the plant was infected, it will show the yellow curve, yellow color and leaf curling and the growth retardation and eventually the severe crop loss. So scientists have always wondering that what's the induced or pathogenic mechanism? What are their mechanism? So now we know that actually this tomato yellow leaf curl virus, they tend to have a small genome. However, it's around like 3 kilobase for each, but they could encode multiple genes to complete their life cycle like DNA replication, cell-to-cell trafficking. To then they survive in the host cells. But there's one interesting feature for virus field. So in here, like the y-axis shows the genome size. So here the viruses. Here is the bacteria, fungi, and here the plant and mammal here. So immediately you could get that actually the virus, they tend to have very small genome size compared to the plants, at least there are 10,000-fold difference. So it has been well known that actually the virus, they tend to have a small genome. However, because to complete their life cycle in host, they have a very high coding complexity genomes. For example, this is SARS-CoV genome. From one single mRNA, they could have multiple open reading frames, and many of them are overlapped. And this is a virus I'm working on. Again, they have many open reading frames are overlapped. So plant viruses is well known for their small genome. However, in order to diversify the gene expression profile to express multiple genes, they have different translation or strategy, like transcription strategy, like produce some subgenomic RNA, or translation like frame shift, or today we focus more on leaky scanning or non-AUG initiation codons. So they could choose different sites to initiate their protein synthesis. So you can tell this is good for virus. I mean, they could have diverse strategy to produce and to help them survival. However, it poses a challenge like the scientists like me. If I want to study their pathogenic mechanism, if I want to study their viral genes, and it will be most difficult if in the beginning we got a new virus species, but we have no idea what genes has been expressed and how do we study their pathogenic mechanism. Okay, so in order to meet this challenge in this project, I decided to use the ribosome profiling that I developed previously to profile the in vivo translation initiation site on viral genome. So if we could have this information, then at least we could have some idea what viral genes are expressed during the infection. Then we could further study their biological function. Okay. So we focus on the tomato yellow leaf curl Titan virus that I mentioned previously, and we profile their in vivo initiation site. So here's the data. So here's the data from the typical RNA sequencing data. So on the top are the virus, the gray box are the annotated virus genes. So you can see that generally we could detect the transcript signal within the genic region. So that's good. Okay, they are expressed. However, it's really hard to tell from this plot to know, okay, this gene is under translation or this gene is under translation, because genes are all overlapped together. But things have become more clear when I focus on our LOTM plot. So we do detect some individual peaks indicate the in vivo initiation site. So now we could more well correlate this in vivo initiation site back to the gene model to know which gene is translated, at least they are expressed during virus infection. So we just go through this profile and identify peak one by one. So in total, actually, you can see we could identify 21 in vivo initiation sites. However, interesting, we found only 4 of them are overlapped with the annotated one. So it indicates that at least in this virus strain, many of them of the initiation site or genes haven't been well annotated. So let's lead to the next question. What are the biological functions of these hidden genes? So that's the next thing we want to know. So actually we studied multiple of them, but in the beginning, in the next, I will just show you one example that we found. So we identified or characterized a novel VV2 viral proteins, which was initiated from the novel initiation site based on our data. So immediately we're wondering, are they really expressed as a viral protein or not? So we quickly just insert the detection tag in the virus clone, and we detect a protein expression during virus infection. So you can see that in either mutation of the novel initiation site, we still could detect a protein, but when we double mutate these 2 initiation sites, protein disappear. So indicated, yes, this VV2 protein are really produced, expressed in the wild type or in a single mutant background. So they are really a VV2 protein. They are expressed during virus infection. But interesting, if you look at this plot again, you will see that when we mutate this second initiation site, that's a 229, they are more dramatically decrease the protein expression compared to the other mutation site. So we go back to check their flanking sequence of the initiation site, because previously people has known there's a well-known, we call it Kozak motif. That's a flanking sequence around the initiation site. They are important to regulate initiation activity. So indeed, we found that this 229 initiation site, they have a flanking sequence more similar to the Cossack. So refer to their initiation activity, what we observed from this Western blot. Okay. So this data demonstrate, yes, they are novel viral protein. They are expressed during infection. So what are their biological function? So we just mutate these two novel initiation sites, and you can see compared to wild type, they show the leaf current phenotype, that's a wild type. But when we mutate this second initiation site, it shows a more flat leaf. So it indicates there is a weaker variance in this mutant strain. So it indicates, yes, they are important and could regulate the virus infection. And of course, we also detect the endogenous virus content from this infected leaf. Yes, the mutant also shows a lower virus content. So that reflects the phenotype we see. And to demonstrate this VV2 are important. Okay, so what are VV2? Actually, VV2 is very tiny. They are less than 100 amino acid protein. So the only prediction structure, function. So we did some protein functional analysis. We want to find out some known motif. But the only one we can find out is the transmembrane domain. So what it did, okay, then we first demonstrate or to review whether they are really in transmembrane domain or not. So what we found out now is that we found this VV2 protein. They are co-localized with the ER. So you can see there's a merge signal here. And also this VV2 also co-localized with the amina blue. So amina blue is the marker for the plasma desmata. That's the hole between the plant cells. So right now we have a hypothesis because from the ER to the plasma desmata, actually there's we call it endomembrane transporting system for virus. When virus will be transported, because virus will have a systemic transporting system from cell to cells. So right now we think that these VV2 are very important. They may play some role to facilitate the virus cell-to-cell trafficking. So that's our current hypothesis. And hopefully in the future, we will have more data to show you how this VV2 involved in facilitate virus or promote virus pathogenicity. Okay. So this is our first story about plant virus that we use our technique to profile the hidden genes on this TYLC THV, that's a Thailand strain. But honestly, I want to share with you, this is not a question I want to address in the beginning. So in the beginning, when I went back to initiate my lab, I want to do something related to agriculture. So I go back to the literature, at least in Taiwan agriculture literature, want to find out what are the important virus problems at that time. Interesting, I found that some literature has talked about in Taiwan before 2000, we do have another dominant virus strain, we call it Taiwan strain, which has been dominant and damaged the tomato agriculture in Taiwan. But soon after 2000, people start to find out another strain, that's a strain I just talked about, the Thailand strain in the field. So from those field report, it indicates actually right now in Taiwan, there are two virus strains, and this Thailand is more dominant one. So this indicates there are some differential pathogenicity between these two strains. The Thailand is more stronger one compared to Taiwan. So I'm interested in the question. So what are the molecular mechanisms under the differential pathogenicity between these two virus strains? So what we do first is that we got a strain and we repeat the infection experiment in my lab. So indeed, you can see the Thailand is more severe strain. They are more dominant. They could induce the symptom occur more earlier. So you can see like leaf curling and the growth retardation earlier compared to the Taiwan strain. So we are happy about, okay, we could repeat the observation from the field. So what are the molecular mechanisms for the differential pathogenicity? So to study this, I make a hypothesis. We want to start from the gene expression profile to find out some difference that could explain the differential pathogenicity between these two strains. So we hypothesize it could be the transcript abundance, some protein abundance, or more interesting, we want to find out some new open reading frame that may involve in this. So again, we just perform this two technique on these two virus chain. So here is our preliminary data that we just analyzed and found. So first we found focus on the total RNA expression level. Interesting, we found that for the Thailand strain. So I should mention earlier, like for this virus strain, actually they have a circular DNA. So their RNA could express from the forward or reverse strand, as you can see here. So their RNA expression actually is higher from the forward strand. Relatively ratio compared to reverse strand. If you look at the Taiwan strand, actually their ratio are not that big. So from this observation, we bring a hypothesis. It's still a hypothesis. Right now we are demonstrating that for this Thailand strain, maybe to highly express these two genes from the RNA expression level could promote their pathogenicity. So right now we are studying what are the difference between their promoter activity and are they really correlate with their pathogenicity or not. So this is one hypothesis from the data. The other is from the ribosome profiling. Again, we could find many novel initiation sites on both genomes. So right now we are also focusing on this novel initiation site. So hopefully we could identify other interesting viral genes that are important for their pathogenicity. Okay. So this is just a quick summary for my second part. We focus on the virus. And through our technique, indeed, we could identify some hidden viral genes on their genome. And they do play some biological role to regulate their pathogenicity. And right now we are also interested in how about different pathogenicity among different plant virus strains. Okay. So now I want to move to my second part. That will be the last part of my talk. So with this observation from the plant and plant viruses, there are so many novel AUG and non-AUG initiation sites on the genome. So, and the next question I always on my head is, how do the ribosome, when ribosome scan on mRNA, how do the ribosome recognize this novel AUG and the non-AUG codon? So imagine along the scanning, there are so many, for example, AUG triplet. How do they determine this AUG triplet but not the other? Not mentioned too, there are so many non-AUG triplets. So that's a question I want to address in the following. So we want to know what are the determinants, what are the rules. So in order to address this, in this project, we decided to use machine learning and prediction biology approach to approach these questions. So when I say like, how do they recognize this initiation site? So now we talk about recognition mechanism. So to determine an initiation site, at least there are two regulatory elements are important. One we call it cis regulatory element. That means that there are some sequence information on the mRNA. And second one is more like trans-acting factor. It could be some protein that also involve regulatory this process. So in the first part, we want to first identify what are the cis regulatory elements on the sequence that facilitate the ribosome recognize initiation site. So in this project, we focus on the thousands of the novel initiation sites that we identified previously. Then, okay, so we focus on the novel initiation site could be the AUG or non-AUG codon. Then for each novel initiation site, we collect the sequence or structural information in their flanking sequence. Then based on these thousands of novel initiation sites and the sequence information we collected, then we use machine learning approach. The goal is to identify the model that based on this sequence information to predict here is the initiation site or not. Okay. So I will I will not go through the detail, but here is our result. So indeed, we could identify good prediction model to predict initiation site. So if we focus on the AUG that located in 5' UTR region, so here the y-axis shows the prediction performance. So you just need to believe me. So here is the random expectation. It's like the coin, you throw a coin, they have initiation site or not. And of course, you want to find a model that are better than random expectation. So generally, we try different machine prediction algorithm. Generally, we do identify good model because their performance is significantly higher than random expectation. they will predict initiation site. And we also could identify model that will predict likely non-AUG initiation site. So we are happy about this. And because once you got a good prediction model, then you could address the next important question. What are the features most important for the model prediction performance? Because those features will give us some information. What are the important sequence? So we just based on the non-AUG prediction model, and we dig out the feature important score. So here the important score means that the higher score means they are more important. They have higher contribution to the model performance. So we focus on the non-AUG initiation model and the top one feature. So each dot is one feature. So the top one feature is PWM. They represent the flanking sequence around the initiation site. So based on this information, we just analyze the flanking sequence around the non-AUG initiation site. And clearly you can see there are some symbols here. It's A and the G in a -3 and +4 position, and they are similar to the known, I just mentioned, the Kozak motif. So it indicates, yes, for this non-AUG initiation site, the Kozak motif are very important, at least at this stage associated with their non-AUG initiation activity. But if you look at the AUG group, there's no such pattern. So what are the determinants to determine AUG initiation site? So we look at the top one features here. So there's a short motif, UCU, they show up. It shows that they are most enriched and they play important role in predict AUG initiation site. So based on this information, we just check several CU-rich motifs in the initiation site upstream or downstream region. Clearly, you can see that UCU-related sequence, they are more enriched in the upstream region. And also, we check the mononucleotide. Again, you can see C is enriched, most enriched than U. They are enriched in the upstream region of the initiation site. And similar pattern was also found, but just less enriched, but still you could find out a pattern here for the non-AUG initiation site. So generally, this is our conclusion. When we focus on this novel initiation site, we found that the CU-rich motifs are all important associated with their initiation activity. But for the AUG initiation site in the gene fibron UTI region, all of them, like Kozak, are not important, but for the rest of the type, the Kozak are important. So right now, this is our conclusion. And since the Kozak, actually people previously have discussed a lot, both in plants and in mammon system. However, in plants, people don't discuss about the CU-rich. I mean, no one discuss what's the relationship from CU-rich motif and initiation activity in plants. So we further want to test, are they really functional? Because based on our bioinformatics, we see this association. So we just pick up several genes. So here's one gene as an example. We fuse the upstream region with a report, and including that novel initiation site we identified, and with the reporter genes. And again, we can see when we just mutate these CU-rich tracks, they show the lower protein abundance, but the mRNA abundance are comparable between mutant and wild type. So indicating, yes, those CU-rich tracks indeed they affect protein synthesis. But I just keep saying about CU-rich tracks, but what sequence are they? So we want to identify, are there any key sequence are important? So we pick up one gene again, and we divided this upstream region into different regions and mutate one by one. And clearly we can see that this region 5 are most associated with the lower protein abundance. Then this is loss of function. We also do the gain of function test that we put multiple this region 5 copy in front of the reporter genes. And indeed, they increase the translation initiation activity. So it's again demonstrate, yes, this CU-rich sequence are important. So what exactly are they? So here's the sequence for the region 5. You can see that there are multiple CU-rich residues within this region. And so what's the mechanism? Why these sequences are important? So right now we have a hypothesis. If you can see here, they could have a very well best pairing with the ribosomal RNA that belong to the ribosome. So right now, our hypothesis is that during the scanning process, this CU-rich motif on mRNA, they may best pair with a ribosome, then allow the ribosome have more time to see their downstream initiation site, so they could start the initiation process. So that's our hypothesis. Yeah, okay. So I run out of time, but I want to quickly mention with the model. So I just show you the first beauty. We could identify informative features to find out novel cis elements. The second beauty is that you could use the model to scan on the genome based on mRNA sequence to identify the potential initiation site. So we just use our model, scan on messenger RNA. We could identify some codon site, although they could not show any in vivo data, in vivo translation activity in our data set. However, they have a high prediction score, indicates they have the potential to be the initiation site. So we pick up some and we test it. Yes, those potential initiation sites really could trigger protein synthesis. So this is second beauty, that you have a model, you could restart, use model, scan on mRNA sequence to find out potential initiation site. So this is the beauty. The third beauty is that I just talked about within a species, identify potential initiation site through a model. Then could we use our model identify from Arabidopsis to predict the initiation site from tomato? So the answer is yes. So what we did is that we reanalyzed the tomato data set and we group and we use another algorithm to identify the potential initiation site, and we group them based on high to low. So you can see for the group with higher potential to be the initiation site, they really have higher prediction score that generated based on our Arabidopsis model. And we could perform this based on Arabidopsis model to predict initiation site from monoculture, including maize and rice. So indicate that once you have a model, yes, we could do some transfer learning to identify initiation site in another model species. And of course, as we mentioned, that the features CU-rich are very important in the beginning in our Arabidopsis model. And when we focus on the initiation site from different monoculture and diculture, again, we could see that these features are also important for those novel initiation sites. So demonstrate again, these features are evolutionary conserved and important. And that's the interesting thing is that when we go back to check the literature, because when I found this data, I say, okay, so they are important. They are important in plants. So we go back to check literature, found actually it's also very important in other eukaryotics. So in human or in human, this is a plant virus, and this is the mammalian virus. They have found that actually they already indicate there are some important CU-rich motifs are important to regulate their translation activity. So our data from the plant part kind of fill the gap to tell you, actually, there are some novel CU-rich motifs. They are very important, not only in mammalian virus, but also in plants, that they regulate gene expression and protein synthesis. Okay. So here I want to share our good news with you. So the third story that I just talked about has been featured in the genome research, select as a cover story. So what we know right now is that we know there's a runway light on the runway that could direct the airplane or pilot to find the correct runway. So right now, through our analysis, we do identify some important cis elements on the messenger RNA that could direct the ribosome to find out the direct translation initiation site. And the cis elements we identify are shared not only across different plant species, but also in human and the viruses. So although the plants, we know plant and human, they have been diverged in very early time. However, they still share some important cis element or regulatory mechanism for the gene expression. So indicate they are the evolutionary importance of this regulatory mechanism. Okay. So now, like from our technique right now, we know that right now we could profile some novel proteins, and we could at least address what are the biological function of them, And we also doing some study like how ribosome determine what is the recognition mechanism. And in the future, we will also continue to use the experimental and prediction biology approach, want to address this more. We aim to not only reveal the coding potential of the genome, we also want to study their dynamics under what stress condition or what protein could be expressed in a condition-specific pattern to reveal the coding dynamics of the genome. Yeah. I run out of time, so last minute. So this ongoing project right now my lab is doing. So for the recognition mechanism, I mentioned about like 6 regulatory elements, but there's another part I mentioned previously. There are some chance-acting factors that could also help the ribosome to determine the initiation site. So one famous is the pre-initiation complex, which contain the ribosome, scanning ribosome, and also some individual eukaryotic initiation factors. So currently we are screening these initiation factors, want to identify what are the trans-acting factors, what are the regulatory proteins that help to recognize the AUG and the non-AUG codon. Yeah. I will skip this. The idea is that, yes, we do identify 2 groups. One group could help recognize the AUG codon. The other group could help identify the non-AUG codon. But this is preliminary data. We are still working on the mechanism, how these 2 groups work separately or jointly with ribosome and the 6 element to regulate initiation site recognition. Yeah. Okay. So this is the last slide. So as I mentioned, right now we are working on recognition mechanism, and also we are interested to profile more novel initiation site or novel protein under different stress conditions in plants. Okay. So in the end, I want to thank people who contribute this project along the way, and also some collaborator like my great collaborator from Xinhong from Sinica. He's a great virologist, like introduced me into this plant virus field. And also my collaborator about machine learning part, and also I have some project about computer learning. Yeah. So thank you very much.
Thank you so much for the nice talk. That was good. So is there anything known about how post-transcription modification of RNA can modify the choice of the initiation sites?
Post-RNA. Yes, some people help me to look at like the mRNA modification. We do have some evidence that in the initiation site there are some modification, but so far I try to compare the annotated one versus alternative initiation site. Right now I see the same pattern. And right now I could not quickly tell you what is the modification we check, but I didn't. So conclusion is that I think mRNA modification are important. Maybe they regulate the structure nearby, but for annotated one and the novel initiation site, they are similar. That's my quick answer.
Yeah, great.